Welcome to Chapter 3, Biological Molecules. In this chapter, we will examine the major classes of biological molecules that biologists need to know about when understanding living systems, especially in the context of chemistry. One thing that you should perhaps review before going into this chapter is the section on water in our last chapter. Quick review, water is a polar compound. It is an excellent solvent because it can form hydrogen bonds due to its partially negative region and partial positive region. Here I have shown a picture of glucose, and notice that it has some regions that are kind of similar to water, these OHs. And on all organic compounds, anytime you see an oxygen and a hydrogen bound together, there's going to be a little bit of polarity there, which is going to increase the ability of that molecule, if it is small enough, to dissolve in water. So this is glucose, dissolves in the blood, and that's why we call glucose, well, blood sugar. The chapter starts off with a look at what is commonly known as mad cow disease. It's also called Creutzfeldt-Jakob disease, or we could pronounce it Jacob, I suppose. And this was quite a surprise to scientists when they first discovered it, because it is a disease that is caused by a protein. So bullet 1 here, it's a non-living infectious disease, meaning it is non-cellular. We'll add that next bracket there. It is non-cellular. It's not a bacteria. It's not a virus. It turns out that it is a protein, and if you ingest enough of this protein, or if another animal like a cow or a sheep ingests enough of this protein, it can get into the brain, and it causes the brain to become riddled with holes. The animal will become disoriented, maybe even aggressive, and it eventually will generally lead to death. And proteins, we know that they are polymers of amino acids. The protein involved in this disease is a globular protein, and scientists were really amazed that a protein is somehow causing a disease. How can a molecule cause a disease? And what they eventually learned is that this protein that seems sort of infectious, protein, I'll abbreviate it there, is that it interacts with other proteins and causes them to become basically disease-causing proteins themselves, and then those proteins go to interact with other proteins and then cause them to become disease-causing proteins as well. And it's just a cascade of increasing the amount of these disease-causing proteins, which eventually lead to this holes developing in the brain. Why carbon is so important in biological molecules. Organic molecules, firstly, the definition is those which contain carbon usually are going to contain oxygen and hydrogen too. And also, if you look up the definition of organic molecules, you might find a different one online, so it is not exactly a hard and fast definition. Another common addition is that organic molecules, yes, they contain carbon, and they are also found in living things, often made by living things, or maybe consumed by living things. Inorganic compounds is those which lack carbon and those that are not found in living things generally. So carbon is said to be often sort of the backbone of organic compounds. Why? It can form 4 covalent bonds. There are other molecules that can form 4 covalent bonds, but we're going to limit our examination to just carbon because it's so relevant in biology. So 4 covalent compounds. Firstly, looking at the right here, this is methane gas. So actually, some decomposers will actually produce methane gas. If you've seen some of those big explosions in the Arctic that occasionally make the news, I believe that is methane produced by decomposing organisms under the Arctic permafrost. It builds up, and if something sparks it, it can even blow up. Kind of neat. Anyway, carbon, 4 covalent bonds. So there would be 1 here in this compound, methane. There'd be another here, and another there. And again, it's just a sharing of 2 electrons. So we've got a sharing of 2 there, sharing of 2 there, 2 there, and then 2 here. That's a covalent compound. So why is 4 so special? Let's take a look at glucose, which we've already talked about a little bit. On the left, that is also glucose. Here on the left, they've actually written all of the carbons. You can see they even number them. We don't have to get into that in this class. So that's carbon number 1, and then carbon number 2 is right here, and then carbon number 3 is right there, and so forth. The 4 bonds are important because not only can carbon bind a molecule in front of it and then behind it as well, it can then have these side chains also. So let's look at the one on the right now, since I haven't drawn on that one. These carbons here, 1, 2, 3, etc., they're linking the ring structure of glucose together. And then in addition to that, they can have all of these side sort of side branches. So there's a hydrogen there, and then there's an oxygen hydrogen right here. And all of these side branches can give additional functions to the molecule. And by functions, I mean the possibility to form additional bonds with other molecules. Over here, I got to apologize. You'll have to download my PPT to look at this. This is oxygen, and oxygen can just make 2 bonds, and one of them is here. The other one's down there where you can't see it. If I drew another one right up here, we've got one available bonding electron here, another one here. So oxygen can bind with just 2 other atoms. For example, maybe I'm going to make up just another one. So maybe it's another oxygen. So another oxygen over here, they're going to share their 2 electrons. And then another oxygen over here, they will also share their 2 electrons. So we can get a chain here, but there are no side groups possible because all of the bonds in the oxygen atom have been filled up. Whereas if we see this carbon here, this carbon can form 4 bonds, so we get the side chains, similar to glucose. And if you're sick of all this drawing on the PPT, remember you can just get this PPT off of Blackboard and take a look at that as you're listening. Functional groups attached to the carbon backbone of organic molecules. So again, on the right, just as a reminder, that is glucose. It forms a ring structure with carbon as the backbone. We're now going to take a look at some of those functional groups that attach to the carbon. Firstly, what a functional group is. A functional group is, for example, this first one, it's called a hydroxyl group, and it is an OH. And we call it a functional group because they tend to have sort of predictable properties. For example, an OH is polar. It says that right here. It's polar, so it can probably form hydrogen bonds with other compounds. It can also undergo dehydration synthesis with other nearby atoms if there's an enzyme around that can make that reaction happen. So functional groups make the chemical reactions that are going to happen here fairly predictable. We can look at glucose here. We see a lot of hydroxyl groups, and I know that that is going to make it more water soluble because hydroxyls are polar. I also know that glucose is going to be able to form bonds by dehydration synthesis with other compounds, such as other glucose molecules, for example. This second one that I've drawn a red line through, there's the red line right here. That one you don't need to know. I'm not going to ask you about it, so don't worry about it. The next one is a carboxyl group. Sometimes it is called a carboxylic acid. If you look at its structure right here, it has a carbon that has a double bond with an oxygen, then a single bond with another oxygen. Notice there is a negative charge on the oxygen that has a single bond, and that's why it is sometimes called a carboxylic acid. There could be an H plus that has just been broken off from this group right here, which is why there's that negative there. Again, you look up a carboxyl group online and you might find that they've drawn it with an H attached there. It's a carboxylic acid because this H can break off and become an H plus in the solution, and that's why it is called an acid. Okay, next one is the amino groups, and these are, for this class, basically on amino acids. They form part of every single amino acid, as well as also there's a carboxyl group on every amino acid as well. That's why they're called an amino acid, because they have a carboxylic acid on them, and they can perform, again, certain predictable chemical reactions. You can look over all of those functions on the right here. I'm not going to read through all of them for you. Sulfhydryl groups. This one I am not going to ask you to memorize it, but I am going to ask you to be aware that when there is a sulfhydryl group on something, it can form certain bonds called, well, we can just call them sulfur bonds. Basically, they can help stabilize proteins in really high temperatures. So some of those extremophiles that'll live in, say, those hot springs in Yellowstone National Park, their proteins would have a lot of these sulfur groups on them that would help stabilize their proteins and stop them from vibrating themselves apart at those really high temperatures. Phosphate groups. This is relevant when we look at ATP. ATP stands for adenosine triphosphate. And phosphate groups are polar, which means they are attracted to water. They can interact with water. And that's going to come back to us when we look at cell membranes, because phosphate groups form part of cell membranes, which gives rise to the structure of the phospholipid bilayer, or the cellular membrane. The methyl group. This is another one that I'm not going to ask you to memorize, but I may mention it. That's called a methyl group. You might often, well, sometimes see in the news this fairly new science called epigenetics. That is whereby a gene can be turned off in one person, and that gene will even be permanently or semi-permanently turned off in their children and even their grandchildren. And that is because we get these methyl groups binding to the DNA, and it deactivates that region of the DNA, meaning the gene is unusable. So I might mention this a bit, but I'm not going to ask you to memorize it. Real quick again, the ones you need to know: hydroxyl, carboxyl, amino, phosphate, and again, this sulfhydryl, not memorized, but I will mention it, so you need to be familiar with it. How are biological molecules synthesized? So remember, we have monomers and we have polymers, and we can take lots of monomers and bind them together by dehydration synthesis to form polymers. When we do that, what happens is one group of molecules, so say this group, I'm looking at A, dehydration synthesis, and this one. This is two monomers that have already been connected to each other. And if there's a hydrogen here, it is going to be removed as part of this process. We have another monomer here. It has a hydroxyl group. And as I said, hydroxyl groups are often involved in dehydration synthesis. This OH is going to be removed, and we're going to combine these. So one H plus another H here, plus the oxygen, is going to lead to the formation of one molecule of water. So for every bond that is formed this way, we will get one molecule of water produced. In summary, dehydration synthesis, written right here, the removal of water to put together. Basically what it means, NH plus is removed from one subunit, a hydroxyl is removed from another, H2O is formed. Hydrolysis is the opposite. Hydro is the root word for water, lysis is the root word for splitting, so water splitting. Here we take, it is literally the exact opposite of dehydration synthesis, so what's going to happen is water is split into an H plus and a hydroxyl, each of which is then added to one of the newly formed subunits. So over here, B, hydrolysis, this molecule of water will be split into an H plus and an OH minus. How is that done? It's done by a class of proteins called enzymes, which basically facilitate chemical reactions in living things. So this enzyme will grab a molecule of water, break it into pieces, and use those pieces to split this bond. And the result, looking over to the right here, is we have broken that bond, and this H plus has been added to one group. This OH minus has been added to the other group. What are carbohydrates? Carbohydrates are carbon and water. That's what those root words mean, carbohydrates. They're composed of carbon, hydrogen, and oxygen, approximately in the ratio of 1 carbon to every 2 hydrogens to every 1 oxygen. That is not a perfect ratio, it is approximate. Okay, monomers and polymers. So firstly, bullet 1 here, it says monosaccharides, that's the monomer. Disaccharides are just 2 monosaccharides that have been put together. And right over here, glucose and fructose, if they undergo dehydration synthesis, again, we're going to remove a hydrogen over here on the left. We're going to remove that one, and we're going to remove this hydroxyl, forming 1 molecule of water, and then this molecule called sucrose, which is a disaccharide. Di means 2, the root word for, or the abbreviation or whatever for 2, disaccharides. And you can look up the fullest picture here in the PPT. But I've put my face here. I don't know. Adds a little personal note to this, I suppose. What else? Glucose. As I mentioned in our introduction here, this is glucose over to the right. It's a molecule we will talk about a lot, glucose. And because it has all of these hydroxyl groups that I mentioned before, it's one of the big functional groups. We'll talk about it again and again. Hydroxyl groups can form hydrogen bonds with water, which make monosaccharides and disaccharides readily water soluble. They will dissolve in water. If you don't believe me, you can go take a glass of water and put some sugar, table sugar, that would be sucrose here, that's even a disaccharide, put it into water and it will dissolve. Different monosaccharides have slightly different structures. This is a topic every introductory course is required to go through, and I apologize that it is somewhat boring. Don't hate the messenger. So DNA, that's kind of interesting. Deoxyribose, this is where the D in DNA comes from, deoxy, and it is a part of DNA, which we will get to later. The other molecule right here is ribose, and ribose is almost the same name as deoxyribose, and they have added in the book, it says note the missing oxygen. That's why it's called deoxyribose. So over here on ribose, on the second carbon, there is a hydrogen there. But with deoxyribose, there is no hydrogen bound here, so it has a slightly different structure and is not part of DNA. Geez, I got to get rid of my face here, don't I? Well, fructose here is shown is in fruit, and if you eat fructose in fruit, it will have to be converted to glucose before your cells can use it for energy to produce the most important molecule, ATP, which makes other things happen, and we really need it. This one over here is galactose and is in breast milk in mammals, and also will have to be converted to glucose in the infant or baby, if we're talking about human beings, to be usable by energy for energy. Just an enlarged picture of what I already mentioned about disaccharides. They are formed by dehydration synthesis, and this one you got to know that glucose and fructose, when they are bound together, we remove the H and the OH, producing 1 molecule of water over here, and sucrose is the end product. This is called dehydration synthesis, and it's how sucrose is produced. Sucrose is the sugar that is in plants. They transport it up and down their stem and all over. And glucose is the sugar that is transported around the blood in animals. If we continue dehydration synthesis long enough with a monomer such as glucose, we'll get a couple of compounds that are of relevance to human beings, then also to plants. Starch is in plants, glycogen is in animals, such as human beings. They act as energy storage molecules. And both of them consist of glucose molecules, of monomers, excuse me. Glycogen is on the right and is more branched, which means there's more ends of the polymer for enzymes to work on. So if you look Let's first look at the molecule on the left. This is starch, and I'll just trace one big long line of it. It branches once here, and then it gets out to the end. Okay, so there's one branch there. So there's two ends. And then if I went over here and just started maybe in the middle, and I'll so branched there once, branched there again, branched again, branched again, a lot of branches. Okay, you see all these branches in glycogen. Glycogen is an energy store in the liver and is having to be broken down constantly between meals to break off these little end bits. And we get, for each one we break off, that's one molecule of glucose. It goes into the blood to supply the body with energy between meals. And so because animals may need to use to have a really rapid access to energy, glycogen, this is glycogen on the right, has more branches because enzymes which break down this polymer into its monomers, which is for energy in animals, they can only work at the ends of these long polymer strands. They can only work at the ends. They can't start anywhere. So if you were an animal that somehow tried to do an experiment of storing energy as starch, you would not have as rapid access to that energy because just in this little region here, we have one end here and another end there. So there's only in this part two places that enzymes could work to break off those glucose monomers for use as energy. Polysaccharides are chains of monosaccharides. Here we're looking at polysaccharides serving a structural function. Cellulose present in plants, in the cell wall, in wood, serves a structural function in wood and in the cell walls of plants. Excuse me, I said the same thing twice. It is undigestible by animals because of its upside down structure. So if we look down here at this molecule, what we can see is that, let's just take a look at this top bit right here, CH2OH, just as a point of reference. We'll call that the CH2OH. It's on the top here, and then look at the next one. It's on the bottom. Then on the next one, it is on the top. On the next one, it is on the bottom. Okay, that's honestly more detail than you need to know. You can simply say every other one is upside down. The significance of that is that that leads to, when you put a whole bunch of these polymers together, just shown two right here, between them form a lot of hydrogen bonds, which means they really stick together, and they stick together so effectively they form these big called fibrils, micro fibers of cellulose, and animals just cannot break them down. If a cow is eating plants and getting energy from cellulose, it's because they have bacteria in their gut that are helping them break it down. The cow itself can't produce enzymes that will break it down. So primarily a structural role in the case of cellulose. Polysaccharides are chains of monosaccharides. In this case, we are looking at the example of an insect's exoskeleton, and the molecule is chitin. It has an extra group around it that just gives it a little bit of the different properties, and it is also largely undigestible. I wanted to make another point on that last slide. Cellulose, although it is undigestible, that is in fact a benefit, because if you eat a lot of something that's got fiber, like sweet potatoes have a lot of fiber in them, they don't get broken down in your stomach or your small intestine, but when they get into your small intestine, because they can't be broken down, they cause water to enter the small intestine, because water kind of gets bound up with it. As a result of osmosis. So water moves into your small intestine, and it causes the small intestine to bloat, which is actually healthy, gives you a sense of you're full, so it may cause you to eat a little bit less, and it also initiates the small intestine to start kind of squeezing. It's called peristalsis. You don't need to know that word, but it stimulates your digestive system to move food along it at a healthy rate. What are proteins? So proteins are polymers of amino acids. And the two examples we're going to look at, there's just two categories, basically. One, enzymes. So enzymes catalyze chemical reactions. So we were talking about these monomers of glucose, they're bound to each other by these bonds, and an enzyme could break this bond by hydrolysis, break it right there, splitting one molecule of water and breaking that bond. They can do a whole bunch of other things as well. Another example, keratin. This is a structural component of all of these pictures below. That really hairy dog, the horn, I believe that's called a bighorn sheep, if I'm correct. Keratin also forms this structure here, as well as silk in spider webs. And the arrangement of the keratin protein determines what type of structure is produced. So they can be arranged in different ways to produce varying structures. Also, the fingernails are made of keratin. Hemoglobin, another protein, transports oxygen in the blood. Actin and myosin, these are the proteins in muscles that are largely responsible for muscle contraction. And then some proteins are also used as hormones, so they go around in the blood and cause widespread responses in the body. Insulin is a very small peptide hormone, protein hormone, basically. As an example, you've probably heard of that one. Also, antibodies. Antibodies are proteins and they fight diseases. Also, other things like snake venom, antibodies would be responsible for deactivating that venom. Proteins are formed of chains of amino acids. So the monomers here are called amino acids. The polymers are called polypeptides or proteins. Amino acids all have an amine and carboxylic acid group, or a carboxyl group, and then also an R group. The R groups determine the properties of amino acids, and the order of amino acids determines the structure and function of the protein. So right over here to the right, this is the basic general structure of an amino acid. It is composed of what we describe as a central carbon. The hydrogen is little talked about, but it's there. And then on the left is an amine group, and on the right is a carboxylic acid group. The significance of these functional groups is that they can join together to form what are called peptide bonds. More on that later. Down here, highlighted in blue, these are all different R groups. So the R group is called the variable group, and it can be essentially infinitely variable. In human beings and most living things, there's just 20 of them, 20 different R groups, so basically 20 different amino acids. Here's one R group, here is another R group, here's another R group, etc. And then a fourth one and a fifth one. Different R groups result in different properties of the protein. Here you've got a sulfhydryl, and that means it can stabilize proteins in some situations. As I mentioned, the amine groups and the carboxylic acid groups, they can be used in dehydration synthesis to form these predictable bonds. So basically, these amino acids here, I'll circle them, we could form bonds here, we could form a bond here, and form a bond here. It's always between a carboxylic acid group and an amine group. So we'll take, let's see, the phenylalanine and leucine as examples. You don't need to remember leucine or phenylalanine, but this is a carboxyl group here, and then this is an amine group here. And they can undergo dehydration synthesis to link amino acids together. Here we have a closer look at dehydration synthesis. What we are going to look at here is two amino acids, 1, 2, and they're going to form a dipeptide. I've written that right here, a dipeptide. Di means two, so two amino acids bound to each other, and the resulting bond that is formed is called a peptide bond. If we keep adding subsequent amino acids, then we start calling it a polypeptide. So many amino acids bound to each other. Let's look here. Circled in blue are the components of the two functional groups, the carboxylic acid and the amino group, that will be removed and produce that one molecule of water. So again, we've got an OH being removed and then an H being removed. Two H's and an O equal one molecule of H2O. And that is shown all the way over here. Then a bond is formed basically between the nitrogen and the carbon. We can see that right here. This bond that is connecting these two atoms, that is the peptide bond. And this can happen over and over and over to form a long chain of amino acids, each with an R group that will lend specific properties to that protein. So now protein structure, and it is based largely on what R groups are included in the polypeptide. There are 4 levels of protein structure. This is a very rapid overview of them, so do not worry about writing all of this down. This is a quick overview, and then I'll go into each one in more detail. 4 levels. Primary, that is just the order of amino acids, and peptide bonds are what join each monomer together. Then we have the secondary structure, and that is basically repeating local structures, and there's just 2. One is called an alpha helix, that is right here. This is a helical structure, kind of a winding around, and then a beta pleated sheet, and that is kind of a sheet, like a sheet of paper that has been had some creases added to it. Again, don't worry about memorizing that right now. Tertiary structure is when these repeating secondary structures, sometimes these aren't actually present, but generally they are. It's when they then fold up. They will fold up and form sort of a 3-dimensional structure, or a blob. We actually say globular if it is a globular structure. And this is when those R groups interact. So depending on what R groups there are, that'll determine the tertiary structure. Hydrophobic R groups are going to orient towards the inside. So again, those R groups are contributing to the overall structure of the protein. Quaternary structure is simply take 2 proteins, 2 separate polypeptide chains, and connect them to each other. Example shown here is hemoglobin. It is in red blood cells, and it has 4 different polypeptide chains connected to each other. So if I just kind of colored them in very roughly, this would be one polypeptide that has tertiary structure. This would be another polypeptide that has tertiary structure, a third, and a fourth, and they're all bound to each other, and that is what quaternary structure is. Closer look at them. Primary structure. Again, now we're zooming in. So amino acid sequence is what is relevant here. Peptide bonds are the only bonds that contribute to holding this thing together. Let's just take a look at this a little more closely. I'm going to circle the R groups here. So there's an R group here, there is an R group here, an R group there, and an R group there. I'm not going to ask you to identify specifically where these peptide bonds are, but you need to know that peptide bonds are present and they're holding the primary structure of the polypeptide together. If I'm using too much vocabulary for you, you can pause and review that vocabulary for now so that it is not too fast. Again, I don't want to make these lectures excessively long because I know that you can pause them at any time and use the book or the PPT to review. Why are there infinite possibilities of polypeptides? So polypeptides and proteins are essentially what do much of the work in a cell. There's a huge number of possible polypeptides because there's 20 different amino acids. We can put them in any order that we could think of. They could be any length. It could be 2, 3, 4. It could be 1,000 amino acids long. And I already mentioned that one. Amino acids can be in every order. Secondary structure. So alpha helix on the left, beta sheet on the right. Hydrogen bonds form between the NH and the CO groups in the polypeptide backbone. These I have included here, the NH and the CO groups, but I'm not going to ask you to memorize them. Your book mentions them, so I have also mentioned them. All I want you to know is that hydrogen bonds hold the secondary structure together. The R groups are not involved in this bonding in any way. So R groups do not determine the structure of the secondary structures. It is just hydrogen bonds. Closer look at alpha helices. So this is the alpha helix I've already shown you. Keratin in nails, this phenotype is the result of alpha helices. Hair follicles are also the result of alpha helices, keratin, and then in horns as well. Keratin has a lot of alpha helices that contribute to its structure. Turns out it's very hard. Beta pleated sheets. They were first discovered in silk using a microscope. The beta sheets are not visible in a microscope. Actually, sorry, let me take that back. This is a picture used using a microscope, and they are really smooth and flat, which gives silk its very smooth texture. Tertiary structure. That is when the polypeptide is going to fold up into a three-dimensional structure. So let me point out a couple of things that this diagram is showing you. Number one, polar side chains here are shown in blue. Polar means that these R groups or side chains are going to interact with water. These are the R groups, side chains, sorry, R groups. I don't like side chains. R groups, polar R groups. They are going to interact with water, so they'll tend to orient outwards. The non-polar R groups are going to be repelled by water. You can think of them as kind of oily. They will be repelled by water. So this polypeptide will fold up into the structure you see on the right, with a hydrophobic core in the middle, shown by this green color there. And then the polar R groups or side chains will orient outwards and form hydrogen bonds with the water. R group interactions. Hydrophilic polar amino acids orient outwards. I mentioned that. Hydrophobic orient inwards into the core. And oppositely charged ions can attract each other and form ionic bonds. For example, I'm going to just add a detail here. If there was an R group right here that had a positive charge, then it could form an ionic bond with another R group here if it had a negative charge. This would form an ionic bond that would help to stabilize the tertiary structure of this protein. Disulfide bridges can also form. So again, I don't even remember the individual amino acids. I believe it is cysteine. So if we had a cysteine amino acid, it's got one of those sulfhydryl groups here. So there's a sulfur there, there's a sulfur here. They can form covalent bonds, which are really strong, and they will help to stop this polypeptide from unfolding. So they help to stabilize the tertiary structure. And again, you do not need to memorize what amino acids have sulfur in them. I simply want you to know that some do, and they can form these sulfur bonds. A disulfide bridge is the correct term, but the main idea. Sulfur atoms can help to stabilize the tertiary structure, quaternary structure as well. Hydrogen bonds help to hold this structure together as well. Practice, can you, looking at this cartoon diagram, describe the structure of this protein based just on this diagram? So pause the video and see if you can describe the structure of it using as much vocabulary as we have just been talking about. Pause it, review. Okay, if you didn't pause it, you're going to lose that chance to review. Now I'm going to tell you how I would describe it. Firstly, this is a protein that has a tertiary structure because I can see that the polypeptide has folded back in on itself. I would also say it has secondary structure. This appears to be an alpha helix. It is an alpha helix. This arrow here appears to be a beta pleated sheet, as does this flat sort of arrow cartoon. That appears to be a beta pleated sheet as well. Does it have quaternary structure? I would say no. And again, this is not an example of an exam question, just a review strategy. There appears to be only one end here that kind of loops around, goes around, and then comes out here. So it looks like there's just one polypeptide here. So not multiple polypeptides binding to each other. There's no quaternary structure. As far as mentioning the primary structure, I would say that primary structure is also present here, and that it is held together by peptide bonds. But that is sort of a kind of a sort of duh answer, because primary structure is always present in proteins. Protein denaturation. I'm going to post a video on Blackboard that offers you a little more detail than you are required to know, but it's nice to have kind of an animation to see this happening. So feel free to look that up in this week's Blackboard folder. Denaturation is when proteins when a protein's normal three-dimensional structure is destroyed, leaving its primary structure intact. So basically, the polypeptide unravels like a ball of yarn unraveling. Heat, high and low pH, and ultraviolet light and high salt concentrations can denature proteins. Dill pickles seen here are in part preserved because of the salty acidic brine, which denatures bacterial proteins. Depending on how you made them, if you also sterilized everything, that would obviously also help keep bacteria out. That's a big part of canning as well. I like canning, by the way. Then in eggs, the change in color when the clear part of the egg becomes white, that is a result of the proteins that are there denaturing, and it is irreversible. It can't go backwards, just like you cannot unfry an egg. So all proteins are like that. You can't undenature them. What are nucleotides and nucleic acids? So we already mentioned this deoxyribose and ribose. They are 5-carbon sugars. 5-carbon sugars. I didn't mention that last time. Deoxyribose is 5 carbons. Ribose is also 5. So let's take a look at this one first. So the basic monomer of DNA is also a polymer. We'll look at that in later chapters. The nucleotide is the monomer, and it is composed of a phosphate, sugar, and base, and there are 4 types of bases in DNA. The base is shown here. The 4 bases are abbreviated as A's, T's, G's, and C's, and basically we get a whole bunch of them in order, and that is what we call a gene, and it instructs the cell to do various things. Over here, this is ATP, adenosine triphosphate, short-term energy storage, and the energy is stored in between these phosphate groups. Why is this a good way to store energy? Well, imagine putting 3 people who utterly hate each other together. They are going to expend a great deal of energy to try to get away from each other. Well, phosphate groups all have a negative charge, and negative charges repel each other. So when this just this one, just this last bond right here, when that is broken, it releases a tremendous amount of energy that the cell can use to get work done, basically. In ATP, this is ribose. And how do I know it's ribose and not deoxyribose? That is because this oxygen here is still present. It has not been taken off. Here on the left, in the nucleotide here, this one, again, right there, no oxygen, so deoxyribose. DNA and RNA, the molecules of heredity, they are nucleic acids. So many of those nucleotides on the left on that last slide bonded together by, again, dehydration synthesis reactions produce polymers called nucleic acids. That's essentially DNA, deoxyribonucleic acid. It is double-stranded and contains deoxyribose, not ribose, and the order of bases is the genetic code. For example, this is a very short code, ATGCT. Really quickly here, the significance of this code is that, I'll draw it a little bigger here, ATG. This is going to code for making a protein, and each group, and let me stop for a moment, this part you do not need to memorize right now, but it might help you to make more sense and meaning of the genetic code here. Each 3 nucleotides, so ATG, codes for 1 amino acid to be produced in the formation of a polypeptide. So the next one is GCT. So that's a different group of 3 nucleotides, and that is going to code for another amino acid, some other amino acid. You get it. I already wrote it once. Some other amino acid. So DNA determines what polypeptides are made in a cell. So if you have and that's why we all have different versions of genes. It's different orders of these bases which cause different polypeptides to be produced, which might mean that one person's hair is a little bit curly, another one's is straight, and etc. RNA, not much to say about that for now. Ribonucleic acid, it directs protein synthesis. It's involved in interpreting this code and this order of bases. What are lipids? So lipids, in short, are essentially fats. That's the shortest way you can say describe them. 2, they are formed by combining glycerol with 1 to 3 fatty acids. So right over here, glycerol. Let's take a look at the functional groups. Right here is a hydroxyl. Right here is another hydroxyl. Right here is another hydroxyl. And those are involved in dehydration synthesis. Fatty acids. At the end of each one is a carboxyl group, and we can remove part of it to form a bond in dehydration synthesis. And again, looking down here, if I can direct your attention there, when we form, there's going to be 3 molecules of water, 1 for each of these bonds, because this is also dehydration synthesis. They are, number 3, mostly carbon and hydrogen, so very few polar bonds. So that's why fat is not water soluble. You pour some vegetable oil into water, it will not dissolve. They are great for energy storage, for waterproofing in plants, and they're a major component of all cell membranes. Okay. 3 categories of lipids. 1, oils, fats, and waxes. 2, phospholipids. 3, steroids. Don't worry about writing all that down right now. First, we're going to go through number 1, oils, fats, and waxes. There it is, oils, fats, and waxes. They contain only carbon, hydrogen, and oxygen. Just as the last example, notice this is the same exact example again. They have roughly double the amount of energy per gram compared to carbs and proteins. They are hugely energy dense. If you want to know why, unfortunately, the answer is a lot of chemistry. In short, bonds between carbon and hydrogen, when oxygen is not nearby, are very high energy. That's more detail than you need to know for now, though. Fats are generally from animals, and oils are generally from plants. Continuing with the first category of lipids, fats, they are solid at room temperature because they all have single bonds. They are thus said to be saturated because they have as many hydrogen bonds as possible. This makes them more dense. Why is this? Let's look at saturated fatty acid structure right here. Notice that each of these carbons has all 4 of its bonds bound in such a way that on its left and its right is a carbon, so that forms the backbone of the chain, and then above and below is a hydrogen. So there are as many hydrogens involved as possible, and this forms a straight structure. So over here, a triglyceride, that's glycerol with 3 fatty acids. Each one of the fatty acids is perfectly straight, so they are just lining up with each other. They're lining up with each other, so they're perfectly packed. They can't be more packed, so that makes them more dense, less water soluble. Number 2, oils. These are liquid at room temperature, and they have double bonds, which produce kinks or bends that lead to them being less dense. If you've heard of hydrogenated fats, you probably know that they're, according to most health professionals, not considered to be too healthy. Hydrogenation would add hydrogens to the double bonds, thus saturating them, and this is generally considered to be unhealthy. So looking at unsaturated fatty acid structure here, mostly saturated. These carbons each have 2 hydrogens on them. But then we have this double bond. So right here, these 2 carbons each could be bound to another hydrogen, but they're not. Instead, they have each used their 4th bond to just make a double bond between themselves and the neighboring carbon atom. This has a couple of consequences. One, it causes the molecule to bend. So it bends downward like so. Number 2, this double bond, because there's a lot of electrons in there, so a double bond would be the sharing of 4 electrons. 1, 2, 3, 4. Wow, that's a lot of negatives in there. So that causes the oil to be a little bit less hydrophobic. That region where the double bond is can interact with water because water has partially positive hydrogens. If you're forgetting why that is, please review the polarity of water. And again, this is just another diagram showing the bent nature of those fatty acid chains in unsaturated fatty acids, fats. Hydrogenation, we can chemically add hydrogens to this, and I believe that's largely done to improve the taste because we just like it for some reason, but it is really unhealthy. So these unsaturated fats, if you saturate them, they a little bit more chemistry than we're going to go into, basically just form unhealthy fats. Moving on with the 3 categories of lipids. Waxes, these are generally indigestible. If you've ever bought a cheese that might be covered in a wax to keep it from going bad, the wax is undigestible. Generally, I believe you can eat them, but don't take my advice for it. Maybe don't eat that wax. What it's used for is water repellent. One example anyway, water repellent on the feathers of birds. It helps them to stay dry. If the bird gets too many breaks in their feathers, then they can't fly. Shown down here, waxy leaves. Leaves do not want water sitting on them because it interferes, firstly, with light. The water will absorb a lot of the light. It won't stop photosynthesis from happening, but it will reduce how much photosynthesis can happen. So the wax helps the water to roll off of the plant. Generally, some plants use that kind of water repellent for other purposes. Category number 2, phospholipids. These are part of the structure of cell membranes. They make up about 50% of the cell membrane. The other 50% is mostly proteins. Okay, firstly, there is a polar head, and that means that this part down there, and the only part I'm going to talk about actually is the phosphate group here because it's polar, means that it's polar so that it can interact with water. It is hydrophilic. It is water-loving. So this part is generally going to orient towards wherever there's water. More on that in a second. Glycerol basically connects everything. So glycerol is 1, 2, 3 carbons, and each one of them had a hydroxyl group that could be used in dehydration synthesis. One of those was used to bind to a phosphate. The other 2 were used to bind to fatty acids here. The fatty acids over here, they are hydrophobic, so they're going to try to orient away from water. Fatty acid tails here. We can simplify the structure of a phospholipid, and this is often done in textbooks, to make it just look like so. And what the circle is the phosphate, and the 2 legs are the fatty acid tails. If I draw a couple of them next to each other, like so, what we would say here is that the negatively charged phosphates are going to be attracted to water. So if there's a bunch of water here, maybe on the outside of the cell, all those phosphates are naturally going to orient towards it, and the fatty acid tails will orient away from it. But wait, isn't there water inside the cell as well? That's where the other part of the phospholipid bilayer comes in. It's actually 2 layers of phospholipids. The other is this one. It's upside down, more phospholipids, and they are upside down. And this is, by the way, just a preview, trying to connect what we're talking about now to how it will be relevant later. This is the other side of the phospholipid bilayer, and in these phosphate heads, again, negatively charged, so they're going to interact, be attracted to the water inside of the cell, and then the fatty acid tails that are all in the middle here orient towards each other, and this structure naturally assembles in water to form membranes. Do not worry about memorizing this later. You can peacefully forget it, but we will come back to it. 3, steroids. Fats are actually part of the hormone system in the human body and also in other animals. Also, they are part of the cell membrane. More on that later. About 2% of the brain. Cholesterol serves as essentially a building block. Cholesterol, a building block for some classes of hormones. Cholesterol, whether you eat any or not, is naturally synthesized in the body, and it's used in cell membranes, part of the brain, and then also to produce hormones such as estrogen and testosterone. And the difference between these two compounds is pretty slight. Looking at them, we can say there is a methyl group, don't have to memorize that name, on testosterone, and there is not a methyl group on estrogen in that location. There's also a hydroxyl group in estrogen, and then a different functional group down here. Don't worry about memorizing these functional groups. I'm just trying to relate them to something a little bit more meaningful. Cholesterol, trans fats, and your heart, and what are you supposed to eat? Holy smokes, is terribly complicated, and if you make a point of reading about it, you'll find that you read a lot of contradictory information out there, and that is because nutritional science is often we don't know all the answers yet. That's why we don't know all of the answers. So some things that we do know. HDL, high density lipoproteins, generally said to be good. They are generally more water soluble, so they're less likely to form a plaque or form a solid kind of a clot buildup on the insides of your arteries and veins. LDL cholesterol, that's low density lipoprotein. What a big word, huh? I'm sorry that there are so many big words in this course. If I could do it another way, I would. But anyway, LDL, generally considered bad, and that is because these, if they're in your blood, they are more likely to lead to cardiovascular disease. They are more likely to wind up as deposits on the insides of arteries and veins. Another point, let me just make this point very simply. All cholesterol has the same structure, all right? Look at that down there, all right? That's cholesterol, and there isn't another cholesterol. So when you say good cholesterol, bad cholesterol, what we are in fact talking about, let me direct your attention to right here, cholesterol. Cholesterol. The cholesterol is inside of this droplet, a droplet of cholesterol, and on the outsides are various proteins and even some phospholipids, and it is the composition of these proteins and phospholipids that determines what kind of interactions it is going to have with your cardiovascular system generally. So what proteins are present here are going to lead to it being good or bad. Again, HDL good, low density lipoproteins bad, and it's because the type of proteins in these droplets that are transporting cholesterol around the body.